Hey U12s, welcome back. So we're going to start today with photosynthesis again. Alrighty. Obviously we've looked at the chloroplast here, so our important parts today are going to be thylakoids and the stroma. Alright, because we're going to be looking at some different stuff within photosynthesis. Now, again, that's the process, and there are stages that we looked at in the last video. So light dependent stage in the thylakoids and a light independent stage, or the Calvin cycle, in the stroma. Okay. And again, this was C3 photosynthesis we looked at. So today's class is going to be on C4 and CAM photosynthesis, the different types. Alrighty, so C3 photosynthesis, obviously we've looked at that. It has a light dependent stage, a light independent stage, or the Calvin cycle. And really it's just to create that glucose, okay? And these C3 plants, they don't have any specialized anatomy. They don't do anything too special, all right? And that's why obviously they work most effectively in that cool and wet environment. All right, but what happens if it gets hot? That's the question here. So again, C3 advantages, they're more efficient than C4 and CAM plants under that cool and moist conditions that I mentioned and normal light level. All right, C3 requires fewer enzymes and no specialized plant anatomy. And most plants on Earth are C3 plants. Okay, so that basic thing that we've looked at. Just recapping Rubisco again, and this is where really the change occurs, okay, depending on if it's cool or warm for these plants. Rubisco here, again, that's ribulose 1,5-bisphosphate carboxylase oxygenase. The important part there, as I've mentioned in class, is the carboxylase and the oxygenase. All right, that is the important part, and specifically the C and the O there, okay? And why is that the case? Because this Rubisco enzyme, really, it can work as a carboxylase or an oxygenase. Now, this plays a big role in the Calvin cycle, okay? And generally, it's going to act as a carboxylase if it's that cool and moist environment, but when it gets hot, the enzyme changes. Okay, so it's a really fascinating enzyme because it can work it can work with both carbon and oxygen. All right, so it has the ability to use oxygen instead of carbons through the Calvin cycle when under high light intensity or high temperature. Okay, and so therefore no carbons are running through here. All right, which means glucose is then not produced, and that's the whole point of photosynthesis for the plant. Okay, so really this is a very inefficient process because you're not producing glucose, but you're still using a bunch of things. All right, all that NADPH, ATP, all of that is going through. Okay, and we call this photorespiration here. Okay, so I just always say photorespiration bad because it's bad for the plant. Okay, it's inefficient. It's it's really a waste here. Okay, and that's the important part. So essentially, how does C4 and CAM plants get around that problem of Rubisco becoming an oxygenase? All right, so we want it to be a carboxylase, so carbon is used in the Calvin cycle, and therefore glucose is made by the plant. All right, so C4 plants have a special structure. All right, so they have specialized anatomy. All right, they use a special enzyme. We don't really need to know the name of that, but they have a special enzyme and they have special anatomy. Okay, what are some examples of C4 plants? Sugarcane, corn, switchgrass, all things that grow in warmer environments. So you can see the C4 plant, and if you're looking at the leaf cross-sectional area, which we're looking at here, it's quite different. Okay, there's no spongy mesophyll there. It's all palisade mesophyll, as you can see in the green. Vascular bundles in a different spot. That's not as important. But what is important is the bundle sheath cells. Okay, and we'll get to that in a sec. So the Calvin cycle occurs in the bundle sheath cells, whereas if we think of the light dependent stage of photosynthesis, that occurs in the mesophyll cells here. All right, so light dependent stage, Oh, that arrow is very difficult to see. Light dependent stage in that mesophyll cell, and then Calvin cycle or light independent in the bundle sheath cell here. Okay, so different cells for the light dependent stage. Essentially, these carbons will travel through the cell wall between these cells through these little holes called plasmodesmata. All right. And again, the biochemical processes are not required for this, but an understanding of the fact that it's got different anatomical structures is very important here. Okay. So what it does is, essentially these mesophyll cells is where gas exchange will occur. All right, so you can see those oxygen molecules here. You can see the carbon molecules here. PEP carboxylase is the enzyme. Again, that's not really important, and this process isn't as important. But anyway, it allows the carbons to be stored into a different molecule and sent through those plasmodesmata, so it can go straight to a chloroplast here, of which it can do the light independent stage of the Calvin cycle in that bundle sheath cell. All right, whereas the oxygen is not allowed through. So in this case, Rubisco has to act as a carboxylase. All right, acting as a carboxylase, and therefore photorespiration, which is bad, doesn't occur. All right, only photosynthesis will occur. All right, so we've looked at the C4 plants now, who of which operate in hot and warm conditions. All right, hot, warm, dry. So now we're going to move on to the last type of plant, which is CAM plants, or CAM photosynthesis. All right, so CAM stands for crassulacean acid metabolism. All right, and these plants are your ones that are going to be living in deserts, right? Extremely dry and arid environments, extremely hot environments. Okay, so very, very little water in these environments. So how does the plant figure out how to photosynthesize and not photorespirate? All right, that's the challenge for these plants. So if we look at the image here, all right, we can see that something's occurring. So it's separating its photosynthesis into night and day. All right. So something to note that we learned in year 11 bio is that the plants have stomata, the openings on the leaves. All right. And those stomata will open during the day so that gas exchange can occur. All right. You need that gas exchange for carbon dioxide so that photosynthesis can occur. All right. That's what we learned in year 11. Now through year 12, we learned that some plants actually do the opposite of that. All right. And why do they do this? Because these plants are in that extremely hot, arid environment with no water. So firstly, they need to conserve water. If the stomata's open during the day where it's really hot, all right, it's going to lose a lot of water. Transpiration is going to increase significantly. All right, and that's bad for these CAM plants. All right. Think of like a cactus as an example. All right, so it actually opens its stomata at night instead, these CAM plants. So as you can see, the carbon dioxide's coming, so gas exchange occurs. All right, carbon dioxide gets changed into some other molecules, so malic acid we're going to think about, so malate and then malic acid. And that malic acid is then water soluble, so it's stored in the vacuoles. So you can see that here. So it's stored overnight in the vacuoles. And that malic acid is obviously very carbon rich because it's made from carbon dioxide. All right. So those stomata are now open. Again, night is cooler, so less transpiration occurs, less water is lost. All right. Therefore, the day occurs. All right. And these plants close their stomata up. All right. So no more gas exchange. But the light is there. All right. So the light dependent stage of photosynthesis can occur. All right, and then when we need our carbons for the Calvin cycle, they are coming from the vacuoles where they're stored as malic acid. That gets converted once again to malate. Biochemical pathways don't need to be known, and then that malate is used as the carbon in the carbon cycle for C4 plants. No, sorry, for CAM plants. Okay. And therefore they save water while still being able to do photosynthesis when it's really hot in the sun there. Okay. So this is the whole picture. Okay, so this is all of them together. So we have C3 plants, which do your regular light dependent stage and carbon cycle. All right, so carbon dioxide goes into the mesophyll cells. Boom. Carbon cycle occurs, glucose created. All right. Whereas C4 plants have different ways to create glucose efficiently in the form of specialized structures. Okay, so they have the mesophyll right next to the bundle sheath cells in a different formation. All right, carbon dioxide comes into the mesophyll cells. The oxygen will come into the mesophyll cells as well through gas exchange, but then the carbon dioxide is the only thing that can go into the bundle sheath cells. So, in other words, light dependent, alrighty. That occurs in the mesophyll cells there, and then the carbon cycle occurs in the bundle sheath cells, which allows glucose to be made efficiently. All right, tick, tick, even when it's hot for these plants. And then lastly, the CAM plants, the desert plants, all right, they use something they refer to as a temporal shift, all right, or a day-night shift, all right, a time shift. So it's not an anatomical thing. It's more just it's a plant behavioral thing, right? If we think of behavioral adaptations. All right, so you can see that here. All right, that temporal shift. Alrighty, and in this case, carbon dioxide comes in at night time where the stomata are now open instead of day. It's stored as that acid, malic acid, so carbon dioxide, and then during the day when the sun comes out, which is our catalyst for photosynthesis, our biological catalyst, there you go. Carbon cycle can occur. Glucose is then efficiently made regardless. Okay. Hopefully this cleared up some questions you guys may have had. All right. And again, I haven't brought out too many videos recently, but I will start to get back on the train and bring out videos every couple of days. Okay? All right, take care. I'll see you soon.